Okay. Okay. Hello and welcome to this scientific meeting. My name is Farzana. I'm the PhD student at the University of Tehran in agrotechnology major. I appreciative of all the researchers, a professor, and a student to join us for this meeting. I want to invite Ms. Zahra Turkaman to host the meet with you.
Hello, my name is Zahra Turkaman. This meeting is organized by the Student Scientific Association of Agronomy and Plant Breeding of University of Tehran. And Professor James Westwood will present on the current status and perspective of parasitic plants research in the genomic era. Professor is an expert in the field of parasitic plants. Before inviting Professor to start the lecture, please don't ask your question until the end of this session. The plan of this webinar is we have a 50 minutes lecture, then we have 10 minutes question and answer at the end of the session. Please write your question in the chat box. We will translate it in English if you cannot translate it. And don't worry if you couldn't be with us until the end of session, because this meeting will record and share on the channel of our YouTube. Professor Westwood, we are so pleased to have you here, and it's a great honor for us to meet you. You can start your lecture.
Okay.
Yes.
Yes. All right. Very sorry for that. That was a computer glitch. I had to reboot the Adobe Connect. So let's get back to where we were, and again, let me know if everything is okay here. You should now see the presentation and hear my voice.
Yes.
Okay, good. Thank you. All right. Well, thank you. Sorry for that little technical hiccup there, but I really want to thank you for the invitation, especially to Nariman, who contacted me and has been so kind. It is very much an honor to speak to you today. Again, I've been working on parasitic plants for quite some time, and I'm really delighted to share some of the things that we've done here, but also a bigger perspective of the field. And I think I was asked to talk about this research in the genomics era, and so I will launch into this. And so, first of all, I want to talk about parasitic plant diversity, and I think you are somewhat familiar, and I'm not sure about this, when I talk to an American audience about parasitic plants. They're generally unaware. Whereas if I talk to somebody from the Europe, Middle East, Africa, there's a greater awareness for parasitic plants. But nevertheless, parasitic plants are a very broad group. And what I'm showing you here is just some of the diversity of the different parasitic plants. And yet it is a tried and true style of living to parasitize another plant. So about 1% of plant species have taken this path towards parasitism as a lifestyle. I'm showing you this slide to emphasize the point, and you may not be able to read the small print there, but what you should notice is that this is a representation of all the plant families, and the ones in red are the ones that have a evolved parasitism. And so you can see that parasitism has evolved multiple times and in different families. And so again, it's the stealing of resources from another plant is commonplace, right? And so I will talk to you today mostly about the Orbancaceae and more about the Convolvulaceae, but I will also mention a little bit about the Malpighiids, the Rafflesias. These ones down here will come up. Most of the reason that we study parasitic plants is because some of them are some of the world's worst weeds. And so here I'm showing you some pictures of plants that I will talk about today. I will introduce the root parasites, Striga and the Orbanci and Philopponci, and spend more time on Cascuda, right? But I have to introduce these root parasites because they are they're important in many ways. They're among the most important of weeds, and a lot of the research that we have done on parasitic plants has been on these species. So you should at least be aware of Striga, or the witchweeds, as probably the most famous parasitic plant in the world. And this one is a huge problem in Africa primarily, although it has escaped to some other regions of the world as well. This pretty plant here you see in the upper left is actually growing attached to the corn crop here, or sorghum or millet, if that happens to be it, but it's attacking a grass crop. And in this field you see at the bottom of the picture, you see where corn should be in that field, and yet what you see instead is a clump of Striga growing around. And the corn, it really is decimated. And so this presents a huge problem, right? When you're talking about already some of the world's poorest people with the least resources dealing with this very difficult parasite. So here you see the Striga. This is on millet, and the Striga plant is here. If you dig up this plant, you find the Striga plant and then the root of the host there. So there's a connection there, a very small connection, but it is enough for the parasite to completely change that host growth. And so you have a lot of very interesting interactions here, which I'll mention in a moment. The other part of this group of the root parasites are the broom rapes, the ones we call the Orbanci and Philopponci species. And these, you can tell me, I think you have these in Iran, but they've kind of divided up the world. They take more of the North Africa, Mediterranean region, and they attack dicot crops rather than grass crops that the Striga attacks. And here you just see some diversity, and I'm just trying to impress you with the devastation there. But here's a picture from a field in Morocco where this is a trial field. So this area infested in front here is Orbanci crenata on fava bean, and just devastating that crop. And one of the things that is so interesting about these families is the communication that happens between the host and the parasite, specifically at the point of germination stimulation, right? So you have the host plant exuding these chemicals into the rhizosphere that signal the parasitic plant to germinate and attack. And this has been one of the great mysteries of parasitic plant research that has been largely solved in recent years. We know a lot more about it. It's very fascinating. And it's not the main point of my talk, but if you're talking about plant communication, plant communication, this is one of the best examples that we have, right? And so just to summarize this, if you're not aware of it, it's been found that this class of chemicals, the strigolactones, are an important plant hormone. They're used within the plant to control plant architecture, but they're also exuded into the environment to facilitate colonization by beneficial fungi, the arbuscular mycorrhizal fungi. And so this is a long-standing symbiotic interaction, but the parasitic plants use that same signal to trigger their germination. And so they can really, the seeds of the parasites sit in the soil and can wait for years and then only germinate when the appropriate host plant grows past it. So it's phenomenal, I think. And to kind of sum up this, I'm going to show you this generalized life history of the Philipponchi and Orobangi species. You know, much of it applies to Striga as well. But the key thing that I just told you about is this point of communication here between that will trigger germination of the parasite. So that's interesting, but it's really just part of the communication that goes on here, because very shortly after the parasite germinates, produces this tiny little radical, then it must form the haustorium. And the haustorium is the structure that makes the linkage to the host root. And that involves a different chemical signaling process, so a very different chemistry as well, but it's also very specialized, or can be very specialized. Then, once there's physical contact, you have cell-to-cell communication, and at this point, the haustorium of the parasite must grow within the tissue of the host and then navigate its way between cells or through cells to reach the vascular tissue and form those connections. And then it must also redirect resources from the host to the parasite and suppress host defenses. So at that point, there's a lot of communication going on as well. And this is the area that I've been particularly interested in this kind of signaling once the two organisms are in a union. And that's what I'll talk about later when I'll talk about Cascuda. And so I'm going to divide my talk into 3 parts. And I've already given you part 1, which is my introduction to the kind of the root parasites, right? Now I'm going to step away from these a little bit, and I'm going to talk about genomics in parasitic plants. And then I'll talk about some specific research we've done with Cascuda, the daughter, and how that fits into this communication business. But let's talk about genomes for a second. And I think that one of the reasons I was asked to talk about this subject is because I've been involved in this for several years now. I was part of this parasitic plant genome project, which we were funded to work on parasitic plants, but we actually never produced a genome from this project. We did transcriptomes, but we worked on it with myself and 3 other colleagues from America, Mike Timko, Claude DePamphlis, and John Yoder, who variously studied different species, and Claude is the molecular evolutionary guy who brings this all together. And the outline of the project looked like this. The framework is that we each had a specialty. My work was in the Philipponchi, Mike on Striga, John on Triphosaria, Claude kind of bringing us together, like I said. But the idea is that these species are all members of the Orobanchaceae, so they evolved from some central point of origin of parasitism, but they have diverged along the way, where you have a transition from facultative to obligate parasitism and the loss of photosynthesis to the extreme end here. So it makes a very interesting framework in which to look at the gene expression. And certainly what we're really interested in was the genomics, the loss and the gain. And so we worked on this for many years. We did some good research, I think, trying to get at these questions. And this is really what we're after in the genomics era here, right? So what are the genomic pre-adaptations that enabled evolution of parasitism? So how do you evolve it in different families, right? In different styles of parasitism? How did the haustorium evolve? And so that haustorium, again, is the essential organ of parasitism. It defines a parasitic plant, right? So, but it's unique. How did it evolve, right? Where did it come from? And what are the consequences then of being a parasite? And I can't give you the answers to all these questions, but I will give you a hint as to where things are going. Because now, within the last few years, we are actually getting parasitic plant genomes, right? And so the field is in this process of rapid acceleration. Some of these have been very, very well-known articles. You may have read them if you're interested in parasitic plants. And I'll just summarize this here with a review table from Yoshida, who put this together recently, where the genomic efforts had been in the world of parasitic plants. And so here you see most of the work has been done on the Orobanchaceae, the rest on Convolvulaceae, and then some other families here. And the way to read this, so here's a lot of RNA-seq transcriptomics has been done, some poor initial surveys of genomes, and relatively few, but some now whole genomes are available. What's also important to think about when you're doing genomics is having functional models that can be transformed. And so what you see here are, we only have transformation for a few species, and those are the facultative parasites, Derospermum, Trifascaria. We've done Philopodkia, but I tell you, it is not easy and hard to reproduce. So give that one a kind of a not so easy. And we've been working on transformation of Cascuda for a few years with only the most modest success. So I would not say that we have that yet. Anyway, that's another conversation. But in terms of sequencing parasitic plants, I put together this table which summarizes really what's been done and what's coming along. So the first ones about 4 years ago were two species of Cascuda, and I'll say more about Cascuda in a moment. And so one of them is very good quality, this Cascuda australis. Then there was the Striga, which is a very important genome. Then the hemiparasite Derospermum, and then this one non-weedy, the sapir, which is related to the Rafflesia that I mentioned. Unpublished, but coming along as a project on an Orobanchi, Cumana, and I've been involved with this. This is actually the work coming out of France from the work of the lab of Stéphane Munoz, with help from other people like Philippe Delaveau. And this is a super quality genome, and hopefully we'll publish this year. But this has chromosome level assembly of the genome of a parasitic plant. And here you can start to really get at some of the questions that are otherwise just hinted at some of these other genomes. So what I want to do for the next couple slides is just present some general conclusions that have been drawn from this work. And getting back to those questions that I was talking about, where did parasitism come from evolutionarily? And one of the things that came out of our work and seems to be confirmed in other parasites is this whole genome doubling that has occurred prior to the evolution of parasitism. So in this case, for example, you're looking at these KS plots, and you can see that the number of genome duplications for Striga is recent, but there's also an ancient one that kind of corresponds to one in the whole family. Mimulus is now grouped to the parasites. So we think that there was a doubling of the genome in these organisms, which then gives you a diversity of extra genes, right? And some of those genes can then be repurposed for parasitism. It gives you the genomic flexibility for that. So that's one common thread. The other interesting thing to think about is then what is lost, right? And so everyone wants to know what's lost, because the parasites that I've shown you so far are very weird-looking plants, right? They are very reduced, often in their leaves. They may have very little root system. The only thing that's truly normal about them really is the flowers, right? So you would expect parasitic plants have lost maybe a lot of genes that aren't needed anymore. Well, yes, that does happen, but maybe not as much as we expect. So here, what you see is this lovely, there's a couple of figures I'm going to show you from this paper by Kai et al. This is, they're the ones who sequenced the saprophyte genome, which is the Rafflesia relatives. And so this is a parasite. I'll say more in a moment. It grows internal to its host. So it's a very different parasite here. But what I want you to notice from this slide is that you have your Striga antheriospermum, right, Orbancaceae. In the yellow, you're seeing a good fraction of missing genes you do not see in the mimulus, right? And with Cascuda, same thing, relative to close its relations, it seems to have lost more genomes. The way you can tell this is having a good quality genome to find the things that are truly missing versus you just fail to pick them up. The saprophyte genome is maybe not the best quality, but it sure indicates a lot of gene loss. And again, this lineage of parasitism is very different from the other two. But let me show you the next figure, which I like quite a bit. This is, again, showing that transition, the gradation from free living to hemiparasite, holoparasite, and endoparasite, right? So even though this saprophyte is a huge flower, it's like as big as a soccer ball, right? A football, you call it, right? And yet the rest of the plant is nothing but a file of cells growing internal to this vine in the grape family. So it's within the host and very reduced, nothing normal about that plant except for really nothing normal at all. The flower is kind of huge and different. But here they're showing you on the scale of degree of parasitism, the amount of gene loss. And there are certain things that you would expect to be lost. And you would expect photosynthesis genes to be lost. And sure, there's been a lot of work done on this, showing that certain chloroplast genes and genes related to photosynthesis are lost as you move toward holoparasitism, and it's extreme in the endoparasite. What's interesting is the loss in some other genes. These defense response genes, why would you lose defense response genes? And ABA, metabolism sensing genes are also lost. So, well, maybe these are related to defense, and maybe a plant that lives within another plant does not need so much defense. But what is striking to me actually is what is not lost here. And so if you look at vegetative development, only a relatively modest 14% of the genes are unaccounted for there. And this is a plant that is very different vegetatively. So what you take from this is a bit of a surprise. So that there are certain genes that seem to be important that you may not think they're important, others that can be lost more easily. And so this is one of the big, at this point, the story in terms of gene loss. The other thing I want to talk about is gene gain, right? So which genes are picked up as a consequence of parasitism. And this is the field of horizontal gene transfer. And we've done some work in this, and primarily led by my colleague, Claude de Pamphilis. But early in our project, we looking across this family again of the Orbancaceae, and we developed this trend here where the more dependent on the host the parasite is, the more horizontal gene transfer events there are. Now again, horizontal gene transfer are genes that are picked up through methods that are not through sexual reproduction, right? So this is more common in bacteria, but in plants it does occur, but quite rarely. Although in parasites, it happens more because you've got that direct physical connection between a parasite and a host. So you can get this transfer of DNA that's picked up and incorporated. And so what you've seen here on the bottom here is just the different species and the horizontal gene transfer events that they share. And of course, you see that there's more in the holoparasites, the obligate parasites. When we added Cascuda to this, you see a similar trend. And here it's presented not by the parasite family, but by the host family, the ancestral host family. So where did they get them from? Striga, for example, we know parasitizes grasses. So Striga's picked up a lot of horizontal gene transfer events from the grass family, right? So this is not surprising. The Philipponchi, mostly from the Rosales, but some other families as well. Cascuda has a much broader host range, and obviously a broader ancestral host range, because it's picked up these genes from a wide variety of plants, and also a lot more, right? So you see how many HGT events are present in Cascuda versus the others. Now, you would think that Cascuda is the champion here, but no. In these days of Olympic athletics, the Cascuda gets the silver medal. The gold medal goes to Sapria, right? So here again, if I regraph all those data, we see that indeed the trend toward more HGT in obligate holoparasites is true, but when you throw in the endoparasite, the endophyte Sapria, it has much more HGT events. So maybe that's not surprising for a plant growing within another plant, that there's even more opportunity to pick up horizontal gene transfer. What's surprising to us is that when we will look, this is before the Sapria data came out, when we were just looking at the horizontal gene transfer in Cascuda versus that in Orobanche, we found that even in these limited numbers of horizontal gene transfer events, there are actually 18 of these events that were in common. So evolutionarily independently, these parasites picked up some of the same genes. So that it tells you something about either the way DNA is transferred to a parasite or the way the parasite makes use of genes. It could be that certain genes are easier to obtain, but also certain ones are more useful. And so it's very interesting to think that there could be very common patterns of horizontal gene transfer in the parasites. So with that, I'm going to wrap up this section of the talk and tell you that in general, from the parasite plant genomes, we can see some of the patterns of the evolutionary history, right? So how they have been changed by the parasitic lifestyle. They seem to be evolving rapidly. They are locked in a relationship with their host that means they have to keep evolving like a pathogen to maintain their position there. And we need more parasitic plant genomes. There are more on the way. This is true of all sorts of plants and all organisms, where the genomes are now easier to get, and so we're going to learn a lot more. But with this, I'm going to turn to my final subject here and talk about Cascuda. And this is a different class of parasites. I've alluded to it and not said too much, but I want to get into the details here. And this is where we see a lot of exchange of materials that might give us a little bit of insight into how parasites are acting. And for this, I'm going to start my video. And it's going, does laser pointer. So turn that off, and I'm going to have you watch this. This is a video made by a former student. It's on YouTube, but it shows you the way this parasite behaves. And so Cascuda, in contrast to the Orbonaceae parasites, it's not germinating in response to a signal, but rather it germinates normally in the spring of the year when other plants are also young seedlings. But then it has to find its host, and you notice that these threads of Cascuda seem to know where the host is. So they may be detecting volatile signals. There have been reports of that. It may be a light quality component as well. But then when it touches the host, it makes that physical contact. And it wraps around, and where it makes tight coils, it will grow a haustoria. And so the haustorium is in the where each of those bumps is. There's a haustorium growing internal to the host and making connections to the host vascular tissue there. And then once it has established itself, it grows, and you can try to pull it off, but it can regrow from those haustoria regions, as you see here. But also, as it establishes, it produces new shoots, and it branches at every node, with each new shoot able to form a new connection to a host. And you get basically a network with one plant making many connections and growing further and connecting to a new plant until you can get really a mass of Cascuda. And with that, you can see this is all growing on a Arabidopsis. So that's just in the lab. And with that, I'm going to keep moving on. So this is just a diagram, what I've showed you there. This is the Cascuda on a Arabidopsis cartoon here, where it's coiled around. If you take a cross section through there, you will see something that looks like this, right, where the haustoria have grown in and are making connections to vascular bundles. And there are multiple types of connections that are made. And as the cells grow through the host tissue, they grow between cells, they can grow through cells, but with cell wall produced, so that you can get actually the situation where you have host cell wall on one side, parasite cell wall on the other, and plasmodist model structure spanning them. So you can get actually a free exchange of cytoplasm and contents that way between host and parasites. You can also get free exchange of water through xylem connections. Phloem connections are less clear because there don't seem to be open sieve pore connections, but there is obviously a high degree of phloem transfer between host and parasite. So there's multiple opportunities for exchanging molecules, as I will talk about in just a moment. But first, I have to show you the connection. So Cascuda is one of those, the few, maybe the only parasite that I know of it from any kingdom that forms connections to multiple hosts at the same time. So here's Cascuda in lentils. See it spreading across down this row. Here's Cascuda attached to one plant, connected to multiple different organisms. So a crop and a couple of different weed species, all connected by the Cascuda. And I want you to think about that as I go through the rest of the talk. And I show you a picture like this. This is from a cranberry field in the U.S., northeast U.S., just covered with Cascuda. It really loves this crop. And so this is an extreme infestation, but it's a total network of plants there. So what's exchanged here? And for this, I'm going to run through a lot of published work and try to just give you a kind of a survey of what's exchanged, and then tell you a quick story about exchange. So we know the water is being exchanged. That's all parasites are after water. We know that sugars and small molecules move. Carboxyfluorocene is a stain of phloem molecules, right? And so you can see that where there's host down here, Cascuda up here, and this is the haustorium. So you can see the the movement of this dye through phloem connections. We know that messenger RNA moves. This is work, one of our most famous bits of work here, where you can actually harvest the Cascuda out here, corresponding to this stem here, and reconstruct completely a host messenger RNA that has moved from the host into the parasite, right? So this is all next generation sequencing, mapping there. And what was astounding about this paper was not only that there's messenger RNAs moving from host to the parasite, but that they go both ways, and that there are a lot of them. So we counted thousands of different genes represented in this transfer. So it was a massive amount of RNA that is being exchanged. And that work, I'll say more about protein later, but there's some early reports many years ago from a German group that this GFP protein is mobile between host and parasite. So proteins, soluble proteins, will move. MicroRNA, this work is also done with a colleague, Mike Axtell, his student at Penn State, came out of our work with the messenger RNA, so we decided to look at microRNAs, and we found out that indeed microRNAs move. And what you're seeing here is a degradome cartoon. Just one of our more interesting. It's a host gene, right? And the parasite makes a small RNA that binds at this point along this gene. It makes a seed element occlusion protein. And what you see is these peaks here represent the degradation of the host mRNA, specifically in the region of that interface, not in the host stem or the parasite stem as much. But what this tells us is that the parasite is specifically able to target host messenger RNAs for shutting them down. Right? And actually this fits into this idea that there is RNA exchange between organisms, right? Especially microRNA exchange. And this all seemed to be a very specialized field for quite a while, except that we've been able to confirm it now in parasitic plants, so we know it happens in plant fungi, plant insect, plant nematode interaction. So it's a common thing in plant pathogen interactions. And now, just recently, late last year, this lovely article in Nature Plants showing that there could be RNAi between plants from exuded microRNAs. And they had this line here to show that microRNAs produced by plants act as signaling molecules affecting gene expression in other nearby plants. So maybe it's a tip of an iceberg of a type of plant-plant communication. But I won't say too much more about that. We've published on that, and Mike has a lot of other good work in progress. The last thing I want to mention is the horizontal gene transfer. Here's Claude. I've already talked about this, so I won't say that, more about that, except to caution, the horizontal gene transfer, that is all inferred as ancient events. We haven't been able to detect real-time DNA transfer. All of these other things seem to happen all the time. Any interaction, you can look at it, it's going on. DNA transfer and incorporation of those genes, it's a different process, different time scale. And so I'm not sure what's going on, but still, nevertheless, things are exchanged. So quickly, what I want to do in the last few minutes is say something about the movement of RNA and protein. And we just recently published this work, actually not this work, this is from the Kragler group in Germany, who gave us the tools, right? And so what they did was they made a fusion of GUS to a tRNA motif, which is sufficient to confer mobility on messenger RNA. So what they were doing is looking into grafting Arabidopsis, so you can have this Gus reporter gene expressed in the shoot, and if you include this tRNA motif, it makes the messenger RNA mobile so that you get it down in the root tip. Right, so a nice breakthrough. And we took that tool and we applied it to our messenger RNA from host to parasite. And just to make a long story short, that we've now just published this work, it does move. So if you take Cascuda and you put it on the host and you cut it like this, here's the host in cross section, the Cascuda in longitudinal section, and you look at it, the construct without the mobility, the tRNA, you don't see blue color here in Cascuda. When you have the tRNA conferring mobility, we do see that. And so we take that to mean that there is translation of a mobile RNA coming from the host, it translated in Cascuda. And then I've got another experiment here where we did the same thing. We had fused it to an ER targeted sequence to further ensure that the protein could not move, even though Gus is not movable. But I'm going to just skip to the main point because it's really the same as I showed you before. Here we've separated the parasite from the host before staining, and what we see is that whenever you have the tRNA motif, you can detect the Gus activity in the parasite and the RNAs as well. So that those are correlated, and so it does look like we're getting that kind of translation, and that got us to look into proteins moving back and forth, and we found that also we see evidence for proteins. Now, where these proteins are coming from, if they're able to move directly or if they're coming as messenger RNA, I don't know, but we do see similar amounts of proteins moving. A little more proteins move this way from Arabidopsis to the parasite versus parasite to Arabidopsis. If we correlate those to our data of mobile RNAs, we have pretty good overlap. About 2/3 of the mobile RNAs have a mobile protein, or mobile proteins have a mobile RNA. So they can actually, depending on the protein characteristics, perhaps move either way. The last thing is just to say, okay, that's interesting, but is there a function for a mobile mRNA or a mobile protein? Because the microRNA story is pretty satisfying because those have been shown to move long distances in plants and have a function, right? But what about RNAs? And this is the work of a former postdoc now in the lab, and this is not published. So this is work on what we call GYP23. When we did our first studies of proteins that were mobile between Cascuda and the host, we found a couple poorly characterized ones that were related to each other. It turns out they had been previously reported as Jasminate inducible proteins of 23 kilodaltons in size, so GYP23. And these were interesting because these proteins are similar to each other. They are encoded in Cascuda, but they're not found in Arabidopsis. And yet we found them in Arabidopsis parasitized by Cascuda. And we looked for their RNAs, and we found that indeed the RNAs of GYP23 are found in Arabidopsis, and they move long distance. So if you look here, if we take the immediate section above the point of contact, we see it. We can still find it out here at the top of the plant in much lower amounts, of course. So this agrees with a lot of our other data on RNA mobility, that it moves and it disappears over time. What's interesting then to think about is, what does GIP23 do? And Soyan, the postdoc, has done a lot of work on that, which I will quickly summarize here. And she did a lot of work, and it turns out that the GIP23 interacts with a protein in the pathway that synthesizes jasmonic acid. It interferes with this CAT2 protein, and she made transgenic Arabidopsis plants either silencing or overexpressing the protein. And the main point here, if you just look at host weight of these plants, there's the knockouts and the overexpressor plants. The knockouts were interesting. We would expect that, and each of these bars is an independent transgenic line. So you have to kind of look at them as a group. Where we've silenced the GIP23 gene, the Cascuda doesn't seem to grow quite as well, whereas where it's been overexpressed, it grows just as well. Plus, overexpressing causes the Arabidopsis to flower a little early. And so the reason we think here it is normal, and here's the overexpressing, flowering early. So with the jasmonate pathway involves a lot of interactions, even relating to flowering. And so it does increase the amount of this FT protein. So just to put that into a model summary here, what we think is going on is that this Cascuda protein moves either as mRNA or as a protein into the host, where it interferes with this CAT2 enzyme, which leads to the production of JA. This has a cascade effect into the JA signaling pathway, which on the one hand could lead to more FT and more early flowering. On the other hand, this pathway is also important in regulating plant defense. So what we really think the parasite is trying to do is probably locally shut down some of these defensive compounds. So that's our working hypothesis right now, and we're following up on that at this moment. But the idea is that here's a very interesting protein that the parasite is expressing, and it seems to have a function. And we're not sure yet exactly what the function is, but it gets into the host, and if you overexpress it, it does affect host physiology. So it may be being used by the parasite as an effector there to like the RNAs, like the microRNAs, to manipulate host growth. And with that, I will wrap it up. These are the final messages here. And what I really want you to do is take home some very simple messages. And that is that some of the world's most destructive and hard to control weeds, and in fact interesting plants, are parasitic plants. And that these plants are experts in communication with their hosts. And so if you're interested in plant-plant interactions, this is a very interesting place to start because of the way the parasites have evolved to communicate on many levels through all sorts of exchanges of chemistry with their hosts. And to top it off, that the microRNAs and messenger RNAs may well be very important molecules in the plant-plant interactions. And with that, I've got a lot of people to acknowledge and too many to list, really. But the people in blue here are the ones who've contributed mostly to the Cascuda project. And here's Soyan, who did a lot of the work at the end there that I talked about. So, and with that, I've hit my time limit, I think. So let's see if we have some time for questions. Hold up for a moment.
Thank you so much, Professor Westwood. Thank you so much for your interesting and impressive presentation. We gain good knowledge in this area. I want to ask the participant, if you have question, there is a hand raise option. You can choose it and ask your question. Please ask your question in English. We have a question in the chat box. It said that with accessible genomic knowledge, can we say that control of this parasitic plant is possible?
Oh, well, that's a great question. So, you know, my younger self would have said yes, right? Because with this knowledge, we have the idea that we can really manipulate the host. We can create resistance. It's much harder than you think. So we've been trying to build on this microRNA work, and some people have shown the ability of host-encoded microRNA to move into the parasite and influence gene expression there. But we've been working on it recently. Several other colleagues have been trying to do it. It's not so easy, because this would be a great resistance mechanism, right? And yet the parasite, for whatever reason, it's not so straightforward. So I think in principle, yes, knowledge is going to give us more tools, more insights. But boy, these parasitic plants are really tough. They're very good at what they do. So yeah, the solution is possible, but it's not going to be easy.
Thank you so much. We have a participant. You can ask your question.
Oh, Professor Westwood, can you hear me?
Yes, thank you.
First of all, thank you very much for your excellent and perfect lecture for Iranian academic science, and I'm so honored that participate in this lecture. I have just a question about secondary metabolite profile of parasitic plants and the way that we can eliminate these secondary metabolites that help parasitic plant to invade host plant. This is an excellent criteria based on your conclusion and given information that you provided for us. I believe that genomics analysis and considering the metabolite profile of parasitic plants can highly affect the integrated management policies for controlling this parasitic plant within the field. So I would like to know, based on your recommendation and based on your excellent and perfect information, how we can affect this metabolite production in parasitic plants that can limit their growth or limit the invasion rate to host plants based on their genomes or the ratio of the genes that may affect the defensive strategies or invasive strategies against host plants. Thank you very much for your kind attention and giving me this opportunity to ask you this question.
Thank you for that nice question. So you are raising a complex subject, which is the metabolism of the parasite. And so we have done a little bit of work with parasite metabolism, and it's frankly, it is harder than looking at RNAs. You don't know where the metabolites have come from, but I will say a couple things to that point. The parasite is controlling much of its own metabolism. So the metabolic profile of that parasite doesn't, I don't know, it doesn't change too much, although they do receive small molecules from the host. And we don't know what that balance is, right? But in a way that parasite does have a lot of control over its own physiology. How do we use this to control the parasites? I'm not entirely sure. Certainly when you get into defense, you would like to be able to have the host plant produce some defenses that are effective against the parasite. But with parasitic plants, because of the plant-plant interaction, you have angiosperm on angiosperm, right? And there is much of the same metabolic machinery. So a plant can't defend itself the way it would against a fungus or a bacteria or something. And so we see fewer examples of good host resistance, and we're still trying to figure those out. I think this will come with more genomics of both host and parasite, and learning more about that interaction. But metabolism, metabolomics, this is a wide open field right now. So again, an interesting topic.
Okay, thank you very much for your kind attention and response that you give me. Really, it was very helpful. And just I have another question, and I am so sorry for the participant that I asked this second question. Another criteria that recently highlighted in some literature is that some researchers suggest that using CRISPR technology, we can introduce new genes to either host plants and parasitic plants' genomes, and by the way, we can reduce their growth ratio. Is it possible in larger scale application to use it as a defensive strategy in fields against this really destructive parasitic plant to control the damages on field? Thank you. If you can considerably give me a little bit of information in this case too. Thank you.
Yeah, very, very good. CRISPR is super powerful, and so in terms of engineering the host for resistance, I would say that there's a lot of potential there. In terms of using it to influence the parasite, it's trickier, and so gets back to my comment about the ability to transform the parasite, right? And so far we're fairly limited in transformation of the most weedy species. I think we'll get this, but what you'd like to do, I think, thinking about maybe moving like a mobile CRISPR element from the host to the parasite, which we've tried to do with very limited success, but we're trying to look at mobile RNAs as a vehicle, right? To get them to produce like a mobile CRISPR in the parasite to do something. Can't say we've had luck yet. Well, we need a lot more time and more money to do that. So theoretically possible. It's not going to. And then it could be done. Will it be done? Will people want it done? Is the question.
Thank you so much. We have another question from Ms. Mahmoudi. Is it possible to control parasitic plants by plant defense hormones like jasminic acid, exogenous application?
Probably not by exogenous application. That's been tried there were some reports of enhancing resistance using a spray of like a salicylic acid inducing chemical. And that was many years ago, and that has not looked like it led to any practical success. JA, no one's shown that either. I think there's some recent work that ethylene is very important in the host-plant interaction, both for the development of the parasite and the host defenses. That may have some promise, but again, in terms of spraying a chemical in the field to do this, we're not there yet. No, there's nothing.
Dear Professor, can you hear me?
Yes, yeah.
We don't have your video.
Oh,
Can you turn it on again?
yeah. I think we had a glitch there. So, all right.
Yes, thank you so much.
Did you hear the end of that question? My response.
Okay, is another one to ask question?
Hello.
Yes.
Can you hear me?
Yes.
First, I have to thank you. This is Nariman speaking. I have to thank you for accepting our invitation for today's webinar. That's great a lot to us. That means a lot to us. And I want just to add some points about your lecture and the questions that some of participants asked. First of all, I think controlling these parasites or entirely controlling these parasites wouldn't be possible. I remember 4 years ago I was reading a paper from Professor Douglas from Cornell University. The paper was about insect pests, in fact, but at the end of the paper there was a sentence that I think was intriguing. Professor Douglas mentioned that controlling insect pests, in fact, is impossible, and there is any permanent or permanent solution for them because they are under a lot of pressure, selection pressure, and they are evolving continuously. And I think parasitic plants are same too, because for example, in the case of Striga or Felipunk or Orbank, they're producing lots of minuscule seeds that can evolve faster even than insect pests. Again, I have to thank you, and I have just one question that I hope you could help me with. What do you think about the Historium? Do you think controlling Historium initiation or Historium attachment to the host plant could be a good target, for example, by gene silencing or, as one of participants mentioned, the CRISPR-Cas system to induce resistance in the host plants? Thank you so much.
Mm-hmm. Yeah. Thanks for that comment. I think, you're right in terms of the evolution of things, and when you come up with a control method, it's of limited use. If you're going to rely on a single control method, then the parasite or the insect or whatever organism you're talking about will get around it. And certainly the genetic potential of parasitic plants is huge. So, maybe, but that said, if we can come up with some different mechanisms, and you know, the idea of the Historium, this is personally an area that I think is really important. I think if you want to create resistance, it's probably the best place to do it, because it's once the parasite has attacked, you've got a connection there. We should be able to figure out a way for the host plant to resist the parasite. I know there's, in the root parasites, there's a lot of work going on in the strigolactone, the germination response area. This is good. We'll probably need multiple mechanisms. That, as per your first comment, I think it can be done. You know, we are learning so much. There's still so much to be learned in terms of these interactions. The people who work on these plants, this is a small group of scientists around the world, relatively speaking to other types of pests. You know, and so I think there's still so much more to be learned in this area, and we will learn how the parasite is able to manipulate the host, and as you can figure that out and counter it from the host side, we will develop some resistance mechanisms at that level. I think the best way to do it is, you know, have it bred into the plant, into the crop, so that you can, you know, plant a resistant host. But you probably need, like you say, better have more than one gene, more than one resistance mechanism, or it will be a short-lived solution. Excellent question.
Thank you so much, dear Professor Westwood. And I really appreciate you for dedicating your time and your excellent presentation. We wish to have more collaborations in the future. And I guess it's the end of this meeting. Thank you so much.
Thank you all for your attention. It was really a pleasure to talk to you.
Thank you so much. Me too. Thank you all participants in this meeting. As I mentioned before, we share this video on the YouTube channel. You can subscribe it and you can watch it on the YouTube channel. Thank you so much and have a good night. Bye.